Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Okay, I'd like to thank the organisers for giving me the opportunity to speak about the Shared Genomics project, which is a project coming out of the Northwest Institute for Biohealth Informatics at the University of Manchester. And it's a project whose aim is to try and engage clinical scientists with infrastructure. So basically we're trying to allow clinical scientists, medics, people working with patients, allowing them to analyse their genetic data. And as I explained, that requires developing an HPC solution or codes that we can parallelise. But along with that, we've got to make the whole access to the whole system and the interpretation of the results easy for our end users. Okay, so the outline of the talk. First of all, I'll sort of talk a bit about the sort of genetic data that we're dealing with, or specifically genome-wide SNP datasets, SNPs, single nucleotide polymorphism datasets, so single changes in a DNA sequence. Genome-wide, so we're looking at changes at many positions within the genome. Then I'll be talking about the actual HPC platform that we've actually developed and how we've gone about trying to make it accessible to our end users. And then lastly, I'll be talking about the process of how we actually also use eScience infrastructure to aid the interpretation of the results, i.e. aid the interpretation of the results we're getting from the statistical analysis run on the HPC platform. Okay, there's that one. Okay, so to outline the scientific problem, our goal here is as the last speaker was mentioned, is to identify the genetic causes of disease. So what we've got here as an example, the DNA sequences of two individuals. One individual who has normal vision and can see the boy catching the baseball, and another person who's got blurred vision as a result of diabetic retinopathy. The two genome sequences of those individuals are largely the same, but there's variations at a particular site. Some of that variation may actually contribute to the reasons why that person with the lower genome has blurred vision. The trouble is, which of those changes is responsible? Which of those changes may be contributing to the disease condition? You can imagine, fairly simple ways in which we could elucidate an answer to that question. We just get the genomes from a load of people with normal vision and genome sequences from a load of people with diabetic retinopathy and compare and contrast. So it's a relatively simple process we can go through to actually try and work out where some of those genetic positions which lead to the disease might lie. The only problem is the scale of the calculations that we're working with. So modern biotechnology allows us to actually study, of order of 500,000 nowadays, probably 1 million different positions in the genome, different loci within the genome. So basically, we're trying to work out which out of 500,000 different positions could be contributing to the actual disease condition. And it's not even that simple, unfortunately. Often, some of the conditions we're looking at or we're interested in may be the result of not just one change in a DNA sequence, but multiple changes acting in concert together or interacting. So if we want to do a systematic analysis of all the possible pairs of changes which might lead to the disease condition, well, if we're starting with 500,000 individual positions, then by the time we're looking at pairs, we're talking of order of 10 to the 11 pairs. So to analyse it in any sort of systematic way, or even a sizable fraction of that 10 to 11 would require a substantial amount of compute power. And then lastly, actually, a lot of the sort of diseases we're interested in, particularly our collaborators are interested in, are maybe the result of gene-environment interactions. The disease condition may not just be due to, maybe have a genetic component, but that genetic component is modified by the environment, the exposures the individual grew up in or exposed to. For example, a particular person may have a particular particular mutation, but if they've also grown up in, say, a dusty household, that may also increase their likelihood of developing asthma. Trying to understand these complex gene-environment interactions and the genetic part of the disease, genetic component of the disease, really requires a domain knowledge of the disease condition. Processing the data really, is going to require compute power. So we've got this difficult sort of situation where we're going to have to develop make use of large amounts of compute power, but the people who are going to interpret the results are going to be people who've got domain expertise, got a detailed knowledge of the actual condition. So what we're going to try and do is allow the people who understand the condition, namely the clinical scientists, to actually directly use the HPC capability that we're developing. Okay, just to reinforce the scale of the task, you can imagine our data might be laid out in some simple data sort of grid, like a spreadsheet, where we got individual genetic positions or SNPs in rows and patients in the columns. The sorts of algorithms we might run to actually identify an individual genetic position might be a relatively simple, low-complexity calculation represented by this inner loop here. For example, it may just be a looping over the patient sample we've got and identifying, doing simple counts of the number of people who've got the disease and the number of people who haven't. So we're talking relatively simple calculations. But on top of that, we're repeating it for, obviously, the number of different genetic locations we're interested in. And then so immediately we've got a much larger calculations. And if we're looking at pairs of SNPs, this is obviously going to be a much larger number. We're talking about SNP pairs here. And then finally, another layer of complexity to add on top of that is that we may actually want to repeat the whole calculation for a number of randomized data sets, permuted data sets, in order to get a measure of statistical significance. So what started out as a very simple calculation here, which may be taking the order of a fraction of a second, maybe then repeated 500,000 times or even larger, and then possibly over many randomized data sets, for example, 10,000 randomized data sets. So we've got a large-scale, compute problem here. But you can immediately see areas where it's ripe for parallelization. The low-complexity calculation here is essentially our single independent task. We can parallelize over the number of SNPs or the number of randomized data sets that we're interested in. And then finally, another area of complexity within the task is we actually need to understand the statistical results. So we can develop an HPC platform to process the data, but if we don't understand the results, then all we're going to do is take large volumes of raw data and transform it into even larger volumes of processed data, which is no use to our end users. So what we actually need to do is we need to add relevant biological information to the data. We need to put those statistical results in a context our end users can understand, and that will enable them to actually make the insights that their domain knowledge will help them to do. So what we're actually going to do is we're going to annotate the statistical results with relevant biological information, such as positions on the genome. Okay. Obviously, the communities which are going to contribute to the sort of work we're doing come in three main flavors, if you like. There's the clinical scientists who actually bring the domain expertise, the knowledge of the disease, and new ways of thinking about analysing the data. They'll suggest ways in which ways of looking at the data that statisticians may not have necessarily thought of. And then we've got software engineers who obviously are doing a lot of the infrastructure development, coding of the HPC analysis codes, and bioinformaticians who are suggesting new algorithms that need coding up, or also suggesting third-party resources that we might use to annotate the statistical results. We can also look at the actual problem in another way, and that's to break it down into the three tasks. So one of the three tasks being, how do we provide a nice, accessible sort of way, interface for our clinical users to access this platform. There's the actual developing of the HPC platform itself and writing of the statistical analysis codes. And then finally, how do we help in the interpretation of their statistical results. And the way we do it is we're going to annotate the results, the statistical results, using web services. We're going to try and automate that process as much as possible. And indeed, that annotation process, throws up metadata, which is, by definition, by being data, is we can also, analyze and do some secondary analysis of. So what I'm going to do for the rest of the talk is sort of go through these individual stages, starting off with the first one, where our goal is to actually provide a simple front end for allowing our clinicians to do HPC on the genetic data. Key requirement, as the last speaker mentioned, is it's got to be easy to drive. So any interface has got to be relatively simple. Just providing the options the user is expecting, i.e., the sort of options which drop down are going to be about the algorithms that they want to apply to the data. All the HPC infrastructure and the bioinformatics tools that we're going to use to annotate the data have to be hidden from the user. The job submission details essentially have to be, all the gory details of doing the HPC actually have to be hidden as much as possible. And then once the analysis is finished, what we're going to try and do, or what we do actually, as this sort of example shows here, is once we've finished the HPC analysis, we load the data into a format that the users are familiar with. So most of our users will be familiar with, say, Office applications, spreadsheets, like Excel, or they'll be familiar with OpenOffice. So what we're going to do is we're going to present the data to them in a spreadsheet grid. So once we've done the statistical processing, what they see, they load up the data and they'll see each result in an individual row, and off the individual row they can right-click and get the annotation data that we've retrieved using the web services. So that's the client that we've developed that our users use, and it talks through to the HPC analysis codes. On the HPC task, what we've actually done is we've taken an existing code base, in this case Plink, which is from Harvard. And essentially we've cleaned it up and then parallelized it. We chose Plink because a number of our users, or some of our users, were already familiar with the code base. It was also written in C++, so it was, it wasn't a difficult step to actually get it into a form where we could easily parallelize it. We've done some cleaning up, for example, removing use of the standard, calls to the standard template library. And then what we've got is we've got a code base which is extremely efficient and got a relatively small footprint. And then we parallelize it by adding in the appropriate MPI calls. We've actually deployed it on our own cluster, which is running Windows HPC Server 2008. Currently, our cluster has 100 cores on it. And actually, throwing cores at this sort of calculation is actually very worthwhile. The problems we're dealing with here are embarrassingly parallel. As the sort of little schematic I had on a previous slide showed, there's a large amount of the there's two areas where it's very ripe for parallelization, and so although it's embarrassingly parallel, you can actually see there's very little communication between processing of different SNPs. That actually means that adding cores yields benefits. So what we've got here is a plot of the speed up gain by adding extra cores to the processing of some test data. The test data has been given to us from one of our collaborators in Manchester, and it's an arthritis data set. And what this shows is the speed up you get as you add the number of cores for different sizes of problems. So what I've got here is processing of 1,000 SNPs in black, processing of 2,000 SNPs in red, processing of 3,000 SNPs in green. This is actually an interaction calculation. So this is what we've actually looked at is all the SNP pairs within those data sets. So if we're looking at 1,000 SNPs, that's of order of 1 million or 500,000 SNP pairs, and so on. And you can see as you add cores, the speed up goes up and then ultimately saturates in accordance with Amdahl's law. But as the scale of the problem increases, the point at which it begins to saturate actually is delayed later and later. So when we're looking at 3,000 SNPs, we've got a good region where it's actually linear. Adding cores is yielding benefits. So by the time we get to 3,000 SNPs, we're talking about 90. An estimate from this data is that our problem is about 99% parallelizable. The parallelizable fraction is about 0.99. So adding cores gives us a 30 times speed up. And to give you an indication of what that represents in real time, on a single core down here, the calculation takes about an hour. So we're speeding that hour calculation up by about a factor of 30. And this is on a single data set. We're not working with any randomized data sets. If I actually wanted to work with say 10,000 randomized data sets as well, on a single core, I'll be talking of order of possibly 10,000 hours worth of CPU time. So what adding cores does for us is it obviously allows us to do calculations on shorter timescales. It also enables new calculations that we wouldn't have even tried before, simply because we didn't have the compute power available to us. And because of that, it enables us to explore the data in new ways. We're not actually afraid of making mistakes, or we're not afraid of actually maybe trying out some hunches on some new ways of looking at the data. Because compute power is now essentially cheap, we can try out a few more scientific hypotheses. Okay. Then on to the last part of those three tasks. I mentioned that one of the key tasks that we see, or one of the things we think is key, is putting the statistical results in context, i.e. we want to annotate the results. We annotate using web services. Why? Well, simply because there's a large number of web services within the bioinformatics community out there already, and it avoids us having to curate large databases of biological information. We can annotate our results. For example, we can find out which, if a particular SNP is in a gene, we can find out what gene what gene that is. We can find out what any putative function of that gene. We can find out that information very quickly. If the underlying information about a gene changes, we're not responsible for that. We're not involved in the curation game. We can just rely upon the providers of the services to do that, to keep their services up to date for us. And as I said before, a lot of services are already collected together in workflows. They already do the sorts of tasks we're interested in. So, for example, Taverna workflows, and therefore we can just use those workflows to do what we want. Just to say about the nature of the annotation data that we're getting back, in a lot of cases it's textual. It's things like publications. It might be finding out the ontology terms used to describe a gene, for example, Go terms. It might be a list of biochemical pathways that a particular gene is involved in. Sometimes it may actually be what I've called here secondary computations. So, for example, we may be interested in providing the user with, if we're looking at a SNP which corresponds to a non-synonymous change, we may be interested in providing the user with an estimate of how destabilizing that change is on the protein structure. So the actual annotation may be the result of a secondary scientific calculation. It may not just be textual data that we're annotating our original statistical results with. Okay. To give an example, what we've got here is a Taverna workflow, which will take an entree gene ID. So we've mapped already from the RS number, the SNP RS number, to an entree gene ID, and then this workflow will map from the entree gene ID to give us a list of pathway. If the SNP's in the middle of a gene, it will give the list of pathways the gene's involved in, and also grab a load of any publications within PubMed that are associated with that entree gene ID. And again, this is a good example of reuse of other people's work. This is a Taverna workflow that we got from a workflow repository, My Experiment, and we just tweaked it slightly to our needs. And what happens is when the user actually loads up a set of results that have come from the HPC analysis, as the results are loaded, the workflow is called and it retrieves the list of publications, and then that list of publications is available to the user under the right click. So the user is actually completely unaware of the fact that the workflow has been executed. All they see is their results in the grid. They right click and they've got a list of publications available to them. The annotation, as I said, is data, and we can do things with that. We can mine the annotation data for patterns. I mean, this is standard practice in bioinformatics. You might look, if you're looking at, say, for example, gene expression data, it's common to look for perhaps enrichment of Go terms, maybe pathways or other functional descriptions or descriptors of your genes. So what we can do this with we can take a very similar approach with the sort of annotation data that we're getting back from the workflows. And again, our goal is to do this automatically. By doing this automatically, the user isn't aware of it, but what we're doing is we're providing with the end user with clues and cues about what the particular SNP the statistical analysis has identified. We're giving them cues about the biological mechanism that might be leading to the actual disease. So we're giving our end users, or helping them with, we're aiding them in interpreting the statistical results. Just to give you a very simple example, what we do with the publications that we get back from that previous workflow that I showed, the Taverna workflow that gets the list of publications from PubMed for us, what we actually do is we take those lists of publications and we feed them into a text mining web service, Termine. And Termine essentially breaks the article titles down into words and then has a go at identifying technical terms within that, assigning a likelihood to each word, the likelihood of it being a technical term. So when we display the publication lists to the user, what we do is we color code the words according to their likelihood of a term, of a particular word being a technical term, and then we also display them in rank order according to the overall term likelihood within the article title. So what we've got at the top is largely articles which are containing words coloured in red and blue, red and orange, and then towards the bottom we've got more articles which are containing words highlighted in blue, which is the lower quartile of technical term significance. If the user wants to, they can click on an individual row and it'll open up the abstract for them as well. So if they want to read a bit more about a particular article, they can click on it, get the abstract, and there'll also be a hyperlink to the actual original source, or at least the PubMed entry. What we've done here is a fairly simple ranking based upon what the Termine web service is giving back to us. But essentially, we could rank in any way we wanted to. We might rank according to the year of the year of publication, the impact factor of the journal, the citation count of the article. Essentially, we're only limited by the services that are available to us. It's up to us. If a service is available, we can make use of it to rank and make suggestions to the user. Okay. What that sort of indicates, that sort of automatic process of sort of suggesting views on the data or suggesting ways in which to interpret the data is what we think is important about the whole platform. What we're trying to do is build a platform which provides thinking space to our end users. It's not just a question of processing data and then providing them with a spreadsheet. What we actually want them to do is actually, we want to aid them in spotting new scientific hypotheses. So we were trying to build a platform that enables them to think, play, and also perhaps share ideas. If you share ideas, then you can communicate them easily to others. So what we want to do is provide a platform that allows for rapid exploration of ideas and then sharing them. And technology such as rich internet applications may provide a means for doing this. So what we've tried to do as an example is make use of a Silverlight component that grabs, also essentially contains shots of the analysis screens that the user generated whilst they were looking at a particular data set. So if I click that, then hopefully what will happen is it will produce a cover flow sequence of images, and what the images consist of is essentially what the user has already looked at whilst they've been inspecting the results in that spreadsheet grid. So that the user's been inspecting the results from the HPC analysis. They've looked at some of the annotations, and now they've suddenly realised, 10 minutes ago there was something they spotted. It jogged something in their memory or sparked something off in their way of thinking. Can they go back and remember what they did? Well, we've actually sort of captured that in visual format with a bit of metadata describing what it was that the user did or what the image consists of. So essentially what we've got here is a visual history of what the user's done. And obviously we can hyperlink. If I can get that one to work, yeah. We can, provide hyperlinks behind the images, and the user can go back to the original source if they want to expand their view. So it may be that the image that they're actually looking at is a good aid memoir, but it's not actually it hasn't got sufficient detail. What we hope to do in the future is actually allow users to edit perhaps some of these cover flows. It may be a situation where you've done half an hour's worth of analysis, you've got 30 or 40 images, a lot of them may not be very productive, and then suddenly you realise you stumble across an idea and you want to communicate that idea to your fellow colleagues, which is the ultimate motivation for this shared genomics project. What you could do is maybe select 4 or 5 images which sort of encapsulate your line of argument, your way of thinking, and then you can send that or allow your colleagues to access that cover flow consisting of just the images that you want them to see. Okay, so if I go back to future plans. Okay, so that's just a sort of a snapshot of what we're trying to develop in the shared genomics platform. Our future plans, well, obviously what we'd like to do is perhaps extend the code base at the back end. So at the moment we've just got the one code base that we've modified, Plink. Perhaps we might use, we aim to reuse some other code bases. For example, our genetics. In this case, probably not. We wouldn't customize in the same way that we'd, customized, Plink to our own ends. Obviously, we want to extend the list of annotations that are available to the user. So again, this will mean making extensive use of workflows which are already in repositories. So if we can go through repositories such as My Experiment and there's a suitable annotation workflow for which will give us information about SNPs, then we can reuse that and make the annotations available to our end user. But I think the real key is perhaps on how we present not only the statistical data but also the annotation data to the end user. Visualization and HCI are going to be key, and that's where the breakthroughs are going to come through. The rich internet applications such as the Silverlight component may help. We're exploring those ideas, but ultimately it's only really going to work if we maintain close collaboration with our end users, because they're the ones suggesting novel ways of viewing the data. So we're trying to maintain a close iterative cycle of exploring ideas and ways of viewing data with our end users. So to end, I will just say thanks to Microsoft for providing some funding. Also our end users who provide us with ideas and allow themselves to be guinea pigs to try our ideas on. So we've got a number of users from local hospitals in the Manchester area. And then finally, thanks to the team in Shared Genomics, Mark Delderfield, the project manager, and Ian Buchan, the director of NIBI, are both in the audience. There's our website. I've got demos on my machine if you want to see that cover flow example, or the actual workbench, the client working. And finally, thank you for listening.
Okay, we have time for a couple of questions. If people could use the microphone, that would be great.
Hi. So one friendly suggestion, if I might, and then a question, which is that the cover flow application you're producing, one of the things that we've seen as important for this kind of creative synergy process is not just being able to look back in time, but being able to juxtapose different parts of time. So the likelihood is it's something they've seen now and something they were doing 10 minutes ago, being if you can put those together rather than having it flipping through, that might help.
Okay. Yeah.
And the second thing along those lines is that Another feature that's often forgotten from software like this that turns out to be very useful is the ability to just randomly spray ad hoc text or notations onto things, particularly if you're attempting to communicate with another party. So it's not these 5 images, it's these 5 images and this annotation about what I think of them that's completely unstructured that turns out to be useful.
Yeah, okay.
So on a related note to that, there seem to be 2 ways generally to make sophisticated systems easier to use. The first is to hide all of the complexity and do it by magic. And the second is to expose the complexity through control to the end users. You seem to have been inclined towards the first, hiding the complexity away. Are there any situations within your tool where you think that exposing user control is possibly the way to go in the future?
Yeah, I mean, as our system grows, then I think we're going to, perhaps hiding the complexity in all cases is going to be difficult. So, for example, as we add in potentially more code bases, or more options of different algorithms to analyze the data, I mean that's one situation where we've got to open up things to the user. It's more as a default. We do try and hide things simply because some of our users are coming from a background where they're not bioinformatics literate, and they're not actually. Some of our users may not actually be that familiar with all the statistical genetics analysis details, and so. We are making some presumptions on their behalf. Also, it allows us in some ways to free up time to focus on the end part, the visualization, which I think is actually the key is where we'll make the biggest gains for them.
And they respond well to that?
So far, yes. But
I think we have time for one more. Okay, in that case, I'll ask. I was wondering, is this software available for other labs to use, or do you have plans to make it available?
Yes, I mean maybe my project manager won't like me speaking. I mean, yeah, we do plan to roll it out. So what we've done so far is just roll it out to in the initial prototype to our sort of local user group, friendly people who are going to give us feedback and maybe tolerate a few more bugs. But we do have plans to release it and make it widely available. Perhaps the biggest issue will be, I mean, at the moment, we've got a client and it connects through to our server which runs the codes. We might not necessarily make that completely open. It may be a question of you email us to request access, and that will go through some process for review. But yeah, ultimately we do hope to have quite a wide international user base.
Okay, thank you very much.